Hello, Dr. Catherine F. Whittington is Assistant Professor of Biomedical Engineering at Worcester Polytechnic Institute, WPI. She started at WPI in January 2018 and runs a research lab that focuses on using tissue engineering principles to develop experimental models of diseases, particularly pancreatic cancer and diseases that affect the lymphatic system. Dr. Whittington was awarded a 2021 Career Development Award from the Pancreatic Cancer Action Network. She is also funded by a Genentech Research Award through the hashtag Fund Black Scientists Initiative and recently received funding through the National Science Foundation and the National Cancer Institute. Dr. Whittington teaches an introductory course in biomedical engineering and an upper-level course in cell and molecular biomedical engineering. In both courses, she works to incorporate more social justice topics related to health disparities and how biomedical engineers interface with members of marginalized communities. She also works with her students to improve their scientific communication skills to help make STEM more accessible to people beyond our classrooms and college campuses. Prior to joining WPI, Dr. Whittington completed a postdoctoral research fellowship at E.L. Lilly and Company in Indianapolis, Indiana. She received her PhD in biomedical engineering from Purdue University in 2012 and her Bachelor of Science in biomedical engineering from Louisiana Tech University in 2006. Thank you.
All right, thank you for the introduction and thank you for the invitation to speak today. My title is a little bit different, but it kind of encompasses the same thing. So we're going to talk about how we in my research lab model pancreatic cancer in the laboratory on the benchtop. We don't do animal studies, although, that's something that we'll likely need to interface with people about, but we don't do those studies. And then as we go through, it's kind of separated into three parts. And so there'll be a couple of spots where I'll pause if people want to ask any questions in the interim instead of waiting all the way to the end. And also there's a couple of spots where I'm going to try to use a Google Jamboard just to get some thoughts from the audience. So we'll see how that goes. It may end up being like in my classroom when I try and it doesn't work as well, but we're going to try that. So we'll get going now. So just really quickly, just kind of an overview of what I'm going to talk about today. I'll talk a little bit about myself and my research group, not too much, but just to give you some context on who we are, why I chose to work on cancer research and why pancreatic cancer research in particular. And then I'm just going to talk about an overview of some of the projects that we do in pancreatic cancer research specifically. We have some other things that we do with other diseases and some things related to tissue regeneration, but I'm just focusing in. So as going along with the theme that you have with personalized medicine, how our work in the future can go towards that, particularly as we think about how we're using our model systems, but currently we're not doing the personalized medicine research just yet. So just really quickly, my research group is located at WPI, of course. We're located in the Life Sciences and Bioengineering building or Gateway. And it's a little bit off of our campus, not too far. Students act like it's in, I don't know, they just act like it's so far away, but it's actually not that far. But my research lab, they've coined us the Dynamite lab. So it's related to dynamic microenvironment and tissue engineering, and you'll get a little bit more of a context of where that's coming from. So they're working on the logo and getting things going so that we have a name beyond Whittington Lab. So just a little bit about myself. I like to share just how I got to where I am. It's a pretty traditional pathway relative to some different pathways that people take, but I like to share that. So I'm originally from Texas and from a smaller town, had never heard of biomedical engineering before I visited Louisiana Tech University on a college visit, thought it sounded interesting and it combined some of the things that I had interest in. So being able to apply science and math and engineering, or what I thought engineering was, to solve medical problems. And so luckily I liked it and it worked out, but I got a chance to do some research as an undergraduate, which was really a great experience for me. But then I decided that I wanted to be a professor, and I don't completely remember why I thought that, but I think just kind of seeing what my professors were doing and working with students, I decided that I wanted to do that. And so I knew I needed to go to grad school to get a PhD. So I ended up at Purdue University and I did biomedical engineering there. I started working in tissue engineering, and I'll talk a little bit about what that exactly is if you're not familiar. But I worked on a project that was related to wound healing and vessel formations or capillary formations, so our tiny blood vessels. And then I decided afterwards that I still wanted to be a professor, so I needed to get more training into a postdoctoral research position. Thought I was going into academia for that, but then I actually ended up at a pharmaceutical company and worked in their cancer cell signaling group. And there I worked on being able, or how we could develop more tumor models that are on the benchtop that could be used for drug discovery. And even though that was a really good experience, I then decided that I still wanted to be a professor, even though I had been in industry for 4 years, and I ended up at WPI. And that's a picture of me when I first got my research lab. So I felt that was pretty cool that I had my own space. And we've expanded and we've added more people. And so it's been fun. And speaking of my people, this is my current research group. We have a little bit of change because some people have graduated and we have some new people this summer, but we haven't taken a group picture all together in a while because we've had different restrictions over the last few years, and then who has the time. But it's a mix of graduate students and undergraduate students. So graduate students are on the top row there, and then our undergraduate students are on the bottom row. They work across the board in all these projects doing active research work. And it's something that I really enjoy doing and try to have as many students in the lab to offer opportunities, although we have limited bandwidth sometimes and we can't take everybody who we like to have. But it's been a great group, and I think we've created a good lab culture for ourselves. So why do I do cancer research? I had no exposure to cancer research before I started my postdoctoral position, but I'll talk a little bit about why cancer and why I use tissue engineering compared to maybe more traditional biology or biochemistry approaches. And so this is a word cloud that I made when I was speaking to our campus Colleges Against Cancer group. I think they're now called the American Cancer Society, like a student chapter. But I made this when I was speaking to them, and it's a word cloud where I just started putting in, all of the people who I personally know or have friends or other family members who know who've been affected. I hate to say it, but if I were to redo this word cloud, it's gotten bigger. People who are affected, what their relationship is to those people, friends, family members, what type of cancers we're dealing with. And as I was going through it, realizing how much this disease impacts people, and it impacts people in different ways, whether you are the patient or you're the caregiver or you're a healthcare provider, there's just so much that still needs to be done in this space, and that we need to make more progress in it. So that's really where I wanted to make an impact. And I particularly started to get more interested in tumor metastasis because it's where the tumor breaks off away from the primary tumor. So I'm thinking about solid tumors, not blood tumors like lymphomas and leukemia, but thinking about solid tumors. Like I said, I work on pancreatic cancer, but the tumor starts to break away, or cells from the tumor start to break away. They go to other parts of the body. And that's really where it's gotten to an advanced stage. And it's really where you see a lot of the cancer deaths or cancer-related deaths coming from is metastasis. And it's because these cells are different. They aren't behaving or reacting or responding to therapeutics in the same way that a primary tumor is. And the primary tumors are usually what our drugs are developed for, not targeting specifically the metastases. So even if you hear that maybe there's a drug for, say, metastatic breast cancer, things are more still geared toward that primary cancer to stop the metastasis from occurring or to slow it, but not targeting where that metastatic tumor is now. So if it's gone to the lung or if it's gone to the brain or to the bone. So needing to work in that space as well. And then when we think about how we actually do drug discovery and develop treatments against tumors, the way we do it in pharmaceutical companies is not really modeling the tumor environment. So if we think about this picture here of, what a tumor could look like, pairing it with this schematic that shows just a little bit more of, you've got blood vessels that are running through the tumor. You've got all sorts of different cell types that are involved. It's not just the cancer cells, but there's all these supporting cells that play different roles. There's immune cells in there. There's also what we call the extracellular matrix that, represented by these little squiggles there, but those are things like proteins and other molecules that are just making up this environment that is supporting this tumor in a way that we don't want it to happen. But it's also very three-dimensional. It's a solid mass. And most of the ways that we do the drug discovery in pharmaceutical companies is that you've got cells that are just sitting on a flat plastic dish that's very stiff. And even though our tumors can be stiff, they're not that stiff, it's plastic. And so you don't get those interactions that you would expect to find in the body that are part of the process of tumor progression and way our bodies respond to the therapeutics. Also, we have the animal models, which there's definitely value in animal models, but in some cases, the animal model cannot fully recreate what the human condition actually is. Some match really well and some don't. Some are as good as you can get. They also can be quite expensive. And the maintenance on that is very different. And so, where could we fit in to try to bridge some of those gaps? Also, when we see some of those mismatches that occur, those can contribute to the fact that we have lots of really promising drugs that start out in the research laboratories and then they don't make it to the clinic for one reason or another. So trying to bridge that gap so we can have more opportunities for effective treatment development. And so I said I use tissue engineering, and a lot of times people are more familiar with tissue engineering for like organ regeneration and tissue regeneration to replace damaged or injured tissues. And I am interested in doing stuff like that with tissue engineering, but also I looked at how, as we go through to try to create these tissues for regeneration, there's utility in being able to use them to model diseases in different ways. And so you've got two different approaches that are shown here. An approach where bottom up, which is more what I do. Well, I guess I do it both ways. But going from different ways that you can assemble things together. So I guess I have some projects where we do that a little bit more, where we're thinking about how you can have cells or cells plus materials or cells plus molecules. That could be proteins, that could be your DNA, RNA, all those types of things can be added together. And on one hand, you can bring these together in different ways and build the tissues, or you can start from the cells and those proteins and molecules themselves. You can add them to materials that have different pore sizes so that cells and things can really get through it. You can have transport of molecules through the material, like you can have oxygen, gas, waste removal, all of that. The cells themselves have the ability to remodel those materials. So they start producing the natural things they would normally do in the body. So then that helps to mimic the structures more like what you would see in the body, to the point where you get something that could be more for regeneration. And so again, we can use these different approaches, and it's very interdisciplinary. So, we're thinking about how can we just better mimic what's happening in the body by what we're putting together on the benchtop. And then why pancreatic cancer research? And so this is where I'm going to give this a shot and see how this goes. So I'm going to put the link in the chat to everyone to this Jamboard. And if you're not familiar with Jamboard, you can just use, I'll pull this over to my screen so you can see it on my screen. But as we look at this, you can use the sticky notes over here to on the left and just type in what you want to add, or you can just create a text box and do it that way. It's whatever you'd like to do, but just really quickly, just put out some numbers, what you think is the 5-year survival rate for pancreatic cancer. Like give people about 30 seconds or so to think about it. Okay, we got some 25s, 10s, 50s, 3, 2, really low, 15. So yeah, so there's a wide range. And, when we think about what actually the survival rate is, and we're thinking about the overall 5-year survival rate. So thinking about once people are diagnosed, these people may or may not be in you're going to have different candidacy when it comes to treatment. But if we go back and we look, thank you everybody for your participation. I appreciate that. When we look, actually, it's an overall 5-year survival rate of about 11.5%. And so I saw some people kind of around that ballpark. It just recently crept into the double digits over the last couple of years. And on the one hand, there's a lot of celebration with it, which is great, but also still that is not great. I saw people were putting, some people have like 30 and 50, 60%. And there are some cancers that have that. There are some cancers that can have up to 80, 90%, but pancreatic cancer, unfortunately, is not one of them. And so if you go to the third slide, if you can advance at the top, but if you go over to the third one, so one of the things is pancreatic cancer is on track to be the second leading cause of cancer-related death. Just give me some ballpark, just quick things on why you think that is. All right, so like maybe 10 more seconds. So we've got people living longer, lack of research, late diagnosis, inoperable cases, the spread, harder to extract perhaps, the connectivity of the organ system, the location of it. So all of these are great thoughts around it, in terms of it, and some of it is what I'm going to talk about with it. But late diagnosis is definitely one of the issues. There aren't really unique symptoms for pancreatic cancer. And so actually I will put these 2 links in the chat for you to go to if you'd like to see some stats on pancreatic cancer and staging. But if people have symptoms of pancreatic cancer, it could be things like fatigue, dark urine, lighter stools, abdominal distress, weight loss. These are all very general things that can happen to people, and they can be symptoms for a whole host of things. So typically people aren't probably going to go right to pancreatic cancer. Also, as people were talking about, there is late diagnosis. And so I'll talk about the late diagnosis piece in terms of where it's located. There's not good screening tools for pancreatic cancer. And so people it doesn't come up in anything. Also, people are talking about better treatments for other cancers. So yeah, if you just kind of balance it out, it's not because more people are actually getting pancreatic cancer. That's actually stayed pretty flat, but it's just some cancers are getting better treatments, and pancreatic cancer research has not advanced to really being able to target it well. There's only a very small number of targeted treatments for pancreatic cancer. If you have a localized, earlier stage tumor, you are more likely to be a candidate for surgical resection. And so that localized cancer, when it's confined to just a primary tumor, roughly about 10% of people are diagnosed at that stage. And then you've got over 82% of people who have either had a local spread of the cancer, so metastasis is already set in, and it's gotten to the lymph nodes, or it's gotten well away. And you see that that distant cancer is actually over 50% of people. And when we look at the survival, the 5-year survival, when you go from that localized tumor that is eligible for surgical resection, you have about 37.4, and then it starts to tank as soon as you get some degree of metastasis. And that's where we get those numbers that go down. So, lots of what you all said is, spot on to what we see. And then somebody even put about access to healthcare, and I'll also briefly mention that in regard to health disparities. And so Black Americans have higher incidence of pancreatic cancer and death rates compared to other people. You can look at the numbers here in terms of incidence. So when the cancer presents itself, the differences that we see there between people of different racial and ethnic backgrounds, and then also the differences in the deaths as well. And I'll put those links in the chat these are from the Pancreatic Cancer Action Network, which is the group that has funded my research. But it's just 2 different articles that talk about some of the different factors related to disparities, looking at some of the risk factors, and I'll talk about risk factors a little bit later, but some of those are more prevalent in the Black community. There's a lot of intersection with all of this as well. So it's not just one thing that's contributing to it, and it's likely a lot more external factors, more so than the biological aspects of it based on current research. But there also are disparities in diagnosis and treatment. So that was mentioned before. And also thinking about where we, how we communicate to people, people's trust in the medical system. Also, a lot of undertreatment is reported. So there's some information in those articles about things like follow-up, and how there's some disparity there. And then the last thing is the lack of representation that actually we have in clinical trials. So even when we're thinking about who is the makeup of the clinical trial populations, that is it's very underrepresented for people from non-white people who are in those, even though the way that the breakdown is for clinical trials definitely does not reflect some of the disease states. This is not just for pancreatic cancers, for other cancers and other diseases as well, but it also doesn't reflect the population of the United States either. So there's a lot that goes into that. And so for my research, I'm thinking about how we can make more effective experimental models that can help progress cancer treatments and understanding of disease so that we can develop better treatment options. And this is one of the spots that I'll pause for questions. And I see one that has come through on the Q&A.
Yeah, and I'll read that too. So, are there any resources that discuss the mismatches of different types of models specifically? Like X mouse strain is good for modeling Y cancer, except for Z conditions, et cetera? Or is that knowledge only available by reading through the journal literature?
Oh, well, always we're going to have, the journal literature is always going to be, step one for a lot of us, especially for like somebody like me who I don't do active animal research. So I'm honestly not as up on the details about the animal models that are out there. But I will say that when you start to think about, I was actually just talking with somebody about another project, and whether or not the mice that we would use for this project would be immune competent, so they would have a full immune system intact, versus immunocompromised mice, which would not have their immune system intact. And so when you go and you're trying to either put in materials or tissues, or even trying to develop a model of a disease, whether or not that immune system is there, we want the immune system sometimes to not be there so we can study what we're doing, but also then it's still in an artificial environment. So you could have situations where you have a mouse strain that is better or worse for something, because maybe you really do need this intact immune system so that you can look at the impact of certain types of therapies, or if it's a disease that is very heavily involved with immune cell populations, to take those out and not being able to repopulate it in the right way may also impact the model. So you definitely will have situations where that plays into it. Also, one of the types of model systems that are used for pancreatic cancer is called the genetically engineered mouse model, where instead of it being you put human cells into a mouse model to form a human tumor, you're actually genetically modifying the mouse to develop like for the mouse themselves to develop the cancer. And so that way you're not dealing with a tissue mismatch at that point. And I see somebody has also put into the Q&A a link related to, I'm assuming related to mouse models and things like that. So thank you. But are there any other questions about what I've talked about so far? And it could also be related to like what I did before, or we can wait for that towards the end. All right, well, if something else comes through, then we will pick it up on the next one. So we're going to go into a little bit about what we do in the lab. So this is just some pictures of students in my lab. I realized that I need to take more pictures. We took this for an outreach activity. And this is just kind of an example of some pancreatic cancer cells that we've used in the lab and combine it with collagen. And I'll talk a bit about why we use collagen. That's what you see with the white in the background. It's just an imaging technique that we are able to use so that we can actually visualize what the collagen looks like and how it's interacting with the cells. So in general, what interests me about tumors, but what interests me about pancreatic tumors specifically. So this is just a schematic showing how you've gone from a primary tumor and then it starts to get a lot of blood vessels. So that's something that fuels tumors with the blood vessels. And sometimes that's used for targeting to try to kind of cut off the supply, but there's varying success depending on the cancers. But then the metastatic process where it starts to break away, the cells start to break away from that primary tumor and start to invade into the tissues and start to get into the blood vessels and lymphatic vessels to start moving and going to other places. So I'm interested in how pancreatic tumor cells are actually invading in fibrotic tissues. I put cancers, I meant put tissue. But that's something that's happening, fibrosis, which is deposited of lots more materials and proteins. I mentioned the extracellular matrix earlier that has a lot of collagen in it in pancreatic cancer. It's also got a lot of hyaluronic acid. And so in fibrosis, more of that gets there and that can impact what's happening in the tumor. So I'm interested in what's happening in that process. I'm also interested in how then the cells are moving into the blood and lymphatic vessels. Particularly, I'm interested in lymphatic vessels because that's a mode of metastasis by the way that the cells are moving into the lymph nodes and then they start to spread elsewhere. It's also an area that's less studied, particularly on the lymphatics themselves. I won't get into the projects that we do on that with this, but it relates to knowing more about what's happening and with fibrotic tissues and lymphatics and then how that impacts. I will briefly talk about it actually in regards to pancreatic cancer, but we also have some work independent of pancreatic cancer. But how both tumor cells and immune cells go through that. And then how fibrosis is impacting pancreatic cancer development when you have certain risk factors present. And that's the work that my grant through the Pancreatic Cancer Action Network is funded by. So just as I mentioned, pancreatic cancer tumors have a lot of fibrosis that causes a really disorganized tissue. So if we look at these tissue sections here that have been stained, you have normal pancreas tissue that looks, nice and uniform. Nothing looks, strange about it. But then you look at the difference between when you take now pancreatic tumor tissue, you see different swirls and the areas where you have a lot more staining showing up in different colors. These are staining different things in the cells. But these swirls in the tissue and the cells relate to that disorganization that comes about, not just from more material being deposited in the space, but then also the cells can start to break down the tissue in areas, and other areas the tissue is more resistant to breaking down. So the remodeling is all kind of out of whack as well. But I'm looking at the fact that it really increases the stiffness of the tissue as well as heterogeneity. And so when we think about heterogeneity, that can mean the way it's organized, the way that the cells are responding, the types of cells that are there. Just a lot of things are going on, and we need to think about how we model that. And so, we're thinking about how both fibrosis can can promote tumor progression as things get worse and worse, but then also too, it can inhibit effective drug treatment or how well the drugs can penetrate and be effective because that tissue has become so dense. And so when we think about it, I mentioned the collagen and hyaluronic acid that are both deposited during the cancer progression and fibrosis. So again, we look at the collagen here is stained in brown, as well as the hyaluronic acid that's stained in brown. These are in separate tissue sections. The blue is representing the cells that are part of the tissue and just other elements, but you have a certain amount of collagen and hyaluronic acid that's in the normal tissue that's adjacent to the tumor. And, it's somewhat uniformity in how it's distributed. But then when we get to the tumor, and if I had a picture of the metastatic site, you look at, there's a lot more of collagen, there's a lot more hyaluronic acid. And the organization scheme of it is very different. So it reflects what I just showed you on the previous slide. And these also relate to poorer prognosis. So these are called Kaplan-Meier curves. And so we're looking at if somebody has low collagen 1 in their tumor, the survival, we're thinking about percent survival over time in this black curve here. But if you've got high amounts of collagen 1, which would represent fibrosis and tumor progression. This gray curve here, for every time point, the survival is lower. So you by the time you're at probably around 6 months, you're lower than 50%, where the other person with lower collagen is around 75. So you can see for both collagen and hyaluronic acid, those gray curves for higher tumor content of these two materials, you're going to have lower survival. So, that's also a problem. And so I'm thinking about how we should be incorporating some of these fibrotic type elements into what I think of as next generation pancreatic cancer models, so that we can understand what is driving that progression of tumors and how these cells are going to be responding to those therapeutics under these conditions that are better mimicking what's actually happening in the body, which is different from some of the ways that we set up these models now, which include those cells sitting on a dish or cells that are maybe exposed to some of these materials, but are not dynamic. As I said before, we're the dynamic microenvironment. So not accounting for some of these changes that happen over time. And so I'm just re-showing this again because I'm thinking about what's the next generation. We should have models that are reflecting the composition of the extracellular matrix. So I'm thinking that they should have collagen and they should have hyaluronic acid in there to reflect those major components of the extracellular matrix that we see in pancreatic tissue. There also should be some type of heterogeneity in there, so that disorganization of the tissue structure. So this is an image of just collagen, type 1 collagen that we find in the body that's been extracted, and we use it in the lab. We can buy this and treat it so that it forms a solid gel or a compliant gel. But this way we can image what the structure of it looks like. But then if we start adding some of the things in there that we find in the extracellular matrix of the tumor, to the right is hyaluronic acid. So we start to see areas where when the collagen is starting to form, it gets bundled in certain areas. So it's more heterogeneous with the addition of the hyaluronic acid compared to the collagen alone. And then if we're also adding another type of collagen called type 4 and another protein called laminin that are also found in the tumors of pancreatic tumors, you again see some differences in the way that the collagen is organizing itself. And so thinking about how we can take those two elements and factor them in so that now we're representing at least that disorganization piece of fibrosis. And then also thinking about this stiffening and the changing over time. So this is just another image of a tissue section of pancreatic tumor, but what the differences are here, it's showing you kind of the staging of it as you go through a normal tumor that is fairly contained. So these would be those patients or normal tissue that is more readily contained. It hasn't maybe not transferred, but hasn't transitioned or transformed into a more malignant cell type. But then they start to form with these lesions that indicate that kind of pancreatic cancer is coming. And so you start to see where the material that's containing the tumor has started to break down, and you start to get things invading out. And this is where you're getting kind of the full-blown cancer formation and going into where, eventually you're starting to get some break off of metastasis. And so I'm thinking about stiffening and how the stiffness changes over time with that fibrosis. And so this is just a way we represent stiffness. It's kilopascals. It's a unit of stiffness measurement. And so we're thinking about, kind of around 1 kilopascal is where normal human pancreatic tissue is sitting. But then as it starts to progress to kind of a more intermediate phase where either early tumor formation or pancreatitis, which is a condition that is a risk factor, you get even more stiffening because you're getting more of that fibrotic activity, more collagen and hyaluronic acid and other things being deposited. So increasing that stiffness, we're gone up to about 2 kilopascals. And then cancerous tumor tissue can go all the way, upwards of 5, 6, 7, 8 kilopascals. So much, much stiffer compared to that normal. But we want to see how to be able to model some of that. And so when we're thinking about the way traditionally people will do this in our field, they'll take cells and combine them with the material. It may be collagen, it may be something else, but they'll take that and they'll put it at a low stiffness, and then they'll have a separate sample and they'll put it at a medium stiffness, and then they'll have another sample and they'll put it at a high stiffness. And that's perfectly fine. You're able to see what the effects of stiffness are on those cells and how they respond. But my question is, well, what about if we are in a situation where we really want to see what's happening as those cells are experiencing changes in stiffness over time, which is what the cells in the body are actually. They're not, sitting in low and they're like, oh, we're going to change over and now we're going to be in high stiffness, because these are also separate cells in these models over here. They don't have any sort of memory, if you want to call it that way, of what they've experienced. And so we're looking at ways that we can actually take a single sample and actually stiffen over time. That's where dynamic stiffening comes in. That's what we're talking about as far as being a dynamic microenvironment lab. And so 2 students, Athenia and Ellie, have been working on using a material. It's called methacrylated collagen, and basically it's been modified so that it can be combined with what we call a photoinitiator. I usually try to avoid saying this photoinitiator. We just call it LAP. But basically what it does, it combines with the collagen. We mix it all together. We heat the collagen because it forms that gel at around 37 degrees Celsius, which is around our body temperature. And then we can expose it to light. And so now that light creates a reaction between the photoinitiator and the groups on the collagen that have been modified, and they start to form more links between them. And that helps to create a higher stiffness of the collagen just based off of exposure to light. We did not develop this. This is an established modification method for collagen that we can purchase. But if we have no light, then we can keep the collagen in the stiffness around like 0.8 to 1.2 kilopascals. That is closer to that normal tissue. And then when we expose it to the light, then we can get it more in that intermediate, kind of closer to that cancerous tissue stiffness, so that 2 to 4 kilopascals is where we can kind of get it, where we expose it to the light. Then we're using a cell line that is just a pancreatic cancer cell line that we again can purchase, that has some, it's been extracted from a tumor and has been established over the years as being reflective of what some of the tumor cell types are like in a pancreatic tumor. But we can take our material from a lower stiffness to a higher stiffness, or we could go even intermediate with it as we do different light exposure regimens. So in this case, Athenia and Ellie have been stiffening it at the very beginning so that cells are just staying at the same stiffness the entire time that we're growing them, or they let them grow for a little bit at that lower stiffness level, so that 800 to 1200 pascals or 0.8 to 1.2 kilopascals. Then they hit it with light part of the way through, and then it stiffens. And so the cells now know what it's like to undergo a stiffening process, and then they're held at that stiffness for the rest of the time. So when we start to stain the cells and look at some of the differences there, so this is just if there's no light, the cells are just at their normal the material is just at its normal stiffness level, that lower one. These cells are stained, the cell body is staining green, and then the nucleus of the cells in the center of the cells is stained blue. So that's what they look like there. If they have undergone this static condition where they've been stiffened from the beginning, they tend to not cluster up in the same way, more single cells. Sorry, I didn't mention the cells are kind of suspended within the collagen at this point. And then when we start to do the progressive stiffening, we start to see some differences with the way that the cells also are clustering and assembling. We're also looking to see like if there's damage to the cells actually during this process. So we've got to look into that. But what we're eventually looking for is how the cells are assembling with each other, and then how do they express certain markers that can give us an indication that it's maintaining similar behaviors to what we would expect to see in pancreatic cancers, and also how things are changing with them. And if they're starting to show any signs of kind of becoming more progressively aggressive. So thinking about like, are they starting to exhibit signs that they might invade or become more metastatic if they were left in culture longer. So risk factors for pancreatic cancer. So we're interested in looking at that. Here's a list of all of the different things that can be risk factors. Some of these things are inherent versus some of these things are environmental. The thing that we are looking at in particular is obesity. And we're trying to figure out why obesity is a risk factor for developing pancreatic cancer. So I put interstitial ECM here. That's just basically the extracellular matrix like I've been talking about that contains the collagen and the hyaluronic acid. Here we're looking at, really how if we put fat cells in that collagen that we've been using, and if we put pancreatic cells or pancreatic cancer cells, how do they talk to each other? What's happening? What's signaling in between the two? And how is that really proceeding when we're thinking about changing those environmental conditions with changing how stiff the material is around these cells? And so our question is what's happening in that crosstalk? How are they communicating with each other? And so one of the things we're looking at is something called exosomes. I guess they're kind of hot right now as far as research is concerned. But it's what are exosomes? They are basically these things called extracellular vesicles. They are produced by the cells. So they're kind of like this little packaged little particle that has different things inside of it. It can be proteins, it can be DNA or RNA, lipids or fats in there, but they have some sort of signaling capacity. And people are looking at what's in there and how is that actually communicating signals between cells. And so we're interested in what's happening with exosomes that are being produced from both pancreatic tissue, pancreatic tumor tissue, and fat cells, and how they're talking to each other. And so we do know some things about it, and I'm not going to go into the details here, but we do know that pancreatic cancer exosomes have some ability to impact what's happening with fat cells, also other cells within that environment. And then exosomes from the fat tissue has the ability to affect inflammation. Inflammation is tied to fibrosis. So again, there are those things that are kind of lining up. And also in cancer, the fat cells can produce to really change how the cells are growing around them, and actually how they're moving and invading. But there's not a model system that really looks at that experimentally and kind of in a systematic way so we can understand what's driving that. And we want to know, is there an impact of this fibrosis in modulating that? Because not everybody who has the diagnosis of obesity is going to develop pancreatic cancer. So why do some people? And is it that environment? Is it the genetic aspects of it? Because there are some common genetic mutations. So does that play into it? You have to have a certain genetic signature plus obesity, plus the way that the environment around plays into it. And does it actually start to contribute to the transformation of these normal pancreatic cells into a pancreatic cancer-like cell in our model? So this is what this project is about, to see how it could actually develop into a into pancreatic cancer. And the last thing is, I mentioned earlier about the lymphatic vessels and how those are impacted, or how they play a role in metastasis. We're interested on how they're being impacted by fibrosis, because people really don't know that, but then how it relates to both the tumor metastasis and also immunosuppression. So pancreatic tumors and other tumors can have a very immunosuppressive environment. So basically, either the immune cells aren't there like they're supposed to be, or they are not functional like they're supposed to be. And so when that happens, you've got this environment that is just not behaving like it should, but the lymphatic vessels they're trafficking tumor cells through it, and they're also trafficking immune cells through it. And so we're wondering is, does the way that fibrotic environment around the lymphatic vessels around these pancreatic tumors change what cells basically enter through, how fast they go through, whether or not they enter, and what's their behavior like when they do enter. So we'd set up kind of a model system where we're looking at things, and I didn't put it on here, but we basically kind of look at it in 2 phases of what's happening here versus what's happening over here in the lymphatic vessels. And this is just an example of a type of system that we would set up where we could actually create these different compartments. That's more of a tumor compartment above. These are just sitting in like a little well. The tumor compartment above, where we can see where there's just signals that are being passed between the 2 cell types. In this case, it's just purely like what's being secreted from either cell type, either the lymphatic cells on the bottom or the pancreatic tumor cells up here in the little insert. Both of them being exposed to stiffening through the materials that we use. Or if they have a different sort of interaction with how they're physically interacting with each other. And so I'll just leave this summary up because I see that there are some questions. And so it's just reiterating. Questions about knowing the influence of the amount. So it's really variable about the amount of collagen and hyaluronic acid. Like, we know that there are increases in the amounts, but it's not necessarily correlated with a specific amount there. The staging of the tumor is going to play a role. Again, it's not just about the tumor cells themselves. There's other cells in the environment called stromal cells. So there's cells called fibroblasts that are really the ones that are putting out collagen. They're the ones really depositing. The tumor cells themselves have some capacity to secrete different types of collagen that have different properties, and so that can impact it. So there's other cells in there. The ability of the cells to break them down is also going to be a factor. But these are some of like the questions on the biology side of it that people are still trying to understand, if there's a correlation between, people have these certain genetic mutations. Does that mean that they're more likely to secrete these collagens compared to, another genetic signature?
Some of the other questions, then let me read them just so our attendees know what's being asked. One is, sorry about that. Sure. Could the exosomal exudates, if that's not redundant, be used for screening for pancreatic cancer?
Yeah, so people are really looking at that as well, not just for pancreatic cancer, but other cancers. Again, it's like understanding what's in that content, and is it consistent or consistent enough to where you can make the connection between either a certain stage of cancer or something else about a population that is significant. And that's where you can get to some of the more personalized medicine type of applications, where you kind of do a workup, not just on the cells themselves or what's being put out there. So that's definitely something that people are looking at, could it be a signature for it? And then you've got the other question that you can ask, Lori.
Yeah. Would you talk more about the specific environmental conditions that are factors in causing pancreatic cancer?
So if we're thinking about the environmental conditions and causing, so when we're thinking about, so you kind of, like I said before, we've got the intrinsic kind of genetic situation where there are, so KRAS is something that's mutated, and it's mutated in about 95% of pancreatic cancers. So that's more intrinsic. When we think about some of the other environmental factors, that's where it's the tougher part, because you saw that list of different risk factors, but it's not like you don't really know why. And some people are looking at, well, is it certain carcinogens that people are exposed to that can be a part of that development? In my case, wondering about like diet and obesity. So I'm looking at obesity itself from just the interaction with the fat cells, but then other people are looking at specific types of diets and how that can play into it. Again, it goes into how the cells are responding and how those types of things might impact the cells to, if it's a spontaneous mutation versus if it starts to shut off certain signals in the cells to where they can't fight off, or the immune side of it, where some of the immune cells are able to be impacted. And so that's a tough question, and that's where people are like actively in the research phase for it. But that really is a tough question about, what's causing the pancreatic cancer in and of itself. And what also makes it tough too is that because we talked earlier about late diagnosis, that you're not always catching it at an early enough stage to maybe do some of the human analysis on tumors to know always like kind of what are those conditions as well. But inflammation, so like obesity is inflammatory. We talked about pancreatitis being a risk factor that also has a lot of inflammation. So I think that there's some things that people definitely look at, in regards to the role of inflammation in kind of transforming those pancreatic cells to where they start to form those early lesions.
That kind of ties into the next question, which was, have any strategies to prevent pancreatic cancer been identified yet?
Strategies to prevent. I'm honestly, excuse me. I'm honestly not as up on that from a research perspective. Again, like you said, that ties into the previous question about understanding what is causing it to begin with. One thing I didn't talk about is that there is a marker called CA19-9 that has been approved for screening for pancreatic cancer. However, only a subset of people are eligible for that screening, because you have to have some familial history and perhaps certain genetic mutations to even do the screening, because that particular marker, even though it's elevated in pancreatic cancer, it's elevated in a lot of things. So on its own, it doesn't do a lot in terms of being able to tell you yay or nay if you are either at risk or if you have pancreatic cancer. It's just not specific enough, and that's one of the biggest problems. So I think that ties into it's because there's just so many things about pancreatic cancer that is not specific, that it's still a matter of trying to figure out where to even go with it, so you can start to look at strategies. So if you look at some of those risk factors, then probably somebody would tell you, okay, it's smoking, watch your diet, lose weight, different kind of exposures. But, beyond that, there's not kind of like a prophylactic type approach to it. Ooh,
great, thank you. One of our committee members asked if you could share where you get your cell lines from.
Oh, well, we currently purchase them from a company called, most of them are coming from a company called ATCC. Again, those are the commercially available lines that, we know if they've come from a primary tumor, if they've come from a metastasis site, which it's usually liver. We know the age of the patient and if kind of what the stage of the tumor was. But yeah, so all of the stuff we knew right now with the lines are very established and not patient related. And then I saw a quick question on there that asked about how to stay up on all the research. It's tough. It is really tough. I depend on my students a lot to be able to, pull in things related to their own projects from research literature. So in the next few minutes, I'm just going to quickly kind of go through where we tie into personalized medicine opportunities, just really quickly. So this is just kind of a schematic that I found around personalized medicine where, you're thinking about how, in my case, I'd be thinking about how we could extract cells from patients. It could be the cells or things that are called organoids that are kind of like little mini tumors in this case that have some of the architecture and the composition of a tumor, but aren't broken down to the individual cellular components. But ideally, we would take those, and if you think about the models we talked about, putting the cells in our models to see how the tumors would be developing in our model systems, and also being able to use our models as a platform for screening to develop an individual approach, which is different from if you were going and doing basically a workup on the patient, understanding their genetic profile, exosome production, perhaps, things like that, which would also be beneficial to what we're doing, but it's not the same line of research that I do. So we would be looking to maybe get information on that to select our patient populations that we would use, but ultimately we would be taking patients, and instead of using our cell lines, we'd swap it out for patient lines and be able to see more specificity and variability between patients. And so I'll just show this really quickly. So when I was a postdoc, we developed a model system that kind of, looked more like a tumor in the sense that you had a tumor compartment that was, we created what we call a little sphere that had tumor cells and the collagen materials, not the exact ones that I'm using right now, but a type of collagen material. And then we could put them into another material. In this case, we were also using collagen to represent the tissue surrounding it, so we can kind of customize both of those compartments. One of the really things that are important when it comes to more personalized medicine and being able to really do more screening is the throughput. So how fast can we go through and actually fabricate and make these models, and how can we analyze them? And so this platform is something that fits into a plate that has 96 wells in it. And so we can create 96 of these little things. And the plate's only about like this big, and it's got 96 wells in there. But then we can create those models in each of those wells. And then what we were able to see is, if we kind of think about the boundary of that tumor that we created, that little sphere, and we can see cells that start to invade out. So we were looking at this as a way to look at invasion potential. So invasion is the kind of the first step of metastasis. So how do these cells actually invade into the surrounding tissue in a way that's more clinically relevant than just like looking to see how they might migrate across a surface. But the cool thing about this is, because we could put in that 96 well plate, we could get a higher throughput higher throughput fabrication analysis. And so this is looking at just a single row in that plate, and we can look at 3 different things in there. And so this is looking at a dosing curve for drugs, where we use a common chemotherapeutic for pancreatic cancer, and looking at 10 different concentrations or doses of it to see how it changes. So if you have a really high, you would expect the cells to, be very responsive, the cells to die, the cells to maybe not invade, things like that. But the nice thing about it is that we could actually, because it's a 96 well plate, we could look at 8 different drugs if we wanted to. And then separately, being able to pull out of just that one single row, we could look at how the cells are growing, how fast they're growing, what their metabolic activity is, so how it's processing things. And also we can look and see how it's actually invading and moving out into that space. And from a personalized medicine standpoint, we would just, as I said, swap out those patient cells instead of cell lines. And then the last thing I just want to mention is a project that I had with senior students who were going to try to incorporate fluid flow. That's another thing that happens in the body. There's fluid that is moving all through the tumors, both through the vessels, as well as just tumor that's fluid that's moving in the spaces between the cells, and it's changing the pressure of the tumor. So that's another thing that you'll hear about, is tumors having higher pressures. But we wanted to incorporate that type of flow and pressures in that way, because that's something that previous model that I talked about didn't have. And then being able to monitor this in real time in the lab. But I wanted to bring this up because this particular project that they were working on, a couple of the students had connection, both personal connection that they had been patients in clinical trials, as well as having family members who were undergoing cancer treatment at the time. And so they wanted to look at it more from a clinical standpoint on how they would be able to use this in a more personalized way, to where instead of using those cell lines, they would use patient samples. And we didn't get to that point with this, but the idea was that their design was done in such a way that they had to think about, if we go and take a patient sample, that means we're taking it from a biopsy that's of tissue that's been broken down into the cells. Then we've got all types of cells there. How do we navigate that? How do we navigate clumps and tissue remnants and things like that? So they had to think about how you would incorporate perhaps a filter that would move. So the idea is that the cells would be housed here ultimately in a material, but then if we want to flow the cells through and actually monitor invasion through that material, you'd have to filter it first and have to deal with that, as well as who's going to be using this. How could you design it so that it's easier for somebody who doesn't do hardcore lab research every day where they're handling this, but how might you do it for somebody who does lab work, but doesn't need a whole lot of steps involved, or needing to troubleshoot anything. They just need to be able to use this. So I just wanted to bring that in because they were thinking about it from a standpoint of how could we personalize this for a patient to where you could put their cells in this housing area here, put a material in this housing area here to represent tissue, flow cells through, account for any sort of filtration that needs to happen from a patient sample. And then could we then, flow through different drugs to see the impact, 3 different drugs, and see how this would impact the patient. Or if you had no drugs, just seeing, what's the consistency between, how much they invade, and is this considered to be a more metastatic tumor versus not. So that's the kind of things that they were thinking about with this project. And then just a few last thoughts is just, we're working to make strides. We're in the early stage. It's interdisciplinary work, but there's a lot of ways that we can approach personalized medicine. And we look to see that personalized medicine is something that's going to be in our future. And I just want to acknowledge the members of my research lab and the different ways that we have been funded. We had a good year this year. We're very grateful for it. But if anybody wants to email me, my contact information is there. I also am on Twitter quite a bit, and I both tweet for myself, our department, and then also for the Black and Biomedical Engineering online community. So I know that we're right at the end, but we did get questions in during, so.
We did, and thank you so much, Catherine. This was incredibly informative, gives us lots to think about, and was a highly engaging presentation. So we appreciate it. Thank you so much.
Yes, thank you. I really do appreciate the opportunity, and I do appreciate the questions and engagement. Appreciate it. It's sometimes tough, but thank you. Just really quickly, I do see something that came in from a panelist. Just a quick thing. If somebody is new to biomedical engineering and would like to learn more about the subject and the issues, what library or other resources would you recommend? So really quickly, I'm a part of the Biomedical Engineering Society. And so that has a lot of resources for students, people who are in the profession. I think they've really been working to revamp like the website and interfaces. They have journal articles that are part of the society. So there's one called the Annals of Biomedical Engineering. There's another one that is, I think it's Cell and Molecular Bioengineering or something like that, where active research is being published. The one on the Annals of Biomedical Engineering is one that is pretty broad. So there's lots of different things. They've recently launched a biomedical engineering education journal that is a part of the society as well. And so that's a great way to see what people are doing in the classrooms in different ways. That was launched, I think, in 2020 or 2021. So I'd say the Biomedical Engineering Society is a good place to start because it is our major professional society. But also just know that there's it's interdisciplinary. And so there's people in mechanical engineering, electrical engineering, chemical engineering who do biomedical engineering research. So even within some of those societies, they have their journals as well that are where people are actively working in those spaces too. So I hope that helps with some ideas of where people who may be interested could go to look as from a library resource standpoint.
Yes, absolutely. Some great leads there for us. So thank you again. And
Yes, thank you. And like I said, if anybody wants to reach out, send me an email. I'm pretty responsive. So thank you.